Having covered the basics of the nervous system, its component parts, its basic function, let's start to zero in on exactly how this action potential stuff works, okay? Let's start by taking you back to your Bio 156. As usual, that was a real prerequisite. So remember that all the cells in your body are surrounded by a cell membrane. There you see on the left-hand illustration an animal cell surrounded by a phospholipid bilayer, the plasma membrane. Why is it called a phospholipid bilayer? Because it's made of phospholipids. So remember, its intent is to separate the inside of the cell from the outside, all right? So there are certain things outside the cell that you might want in, other things you might not want in. Likewise, the cell makes certain products that you might want to leave, but you might not want to leave. And so you have to separate the inside of the cell from the outside of the cell. Cell membranes are made of phospholipids. Remember, that's a lipid, a fat. You can see on the right-hand side of that illustration the little blue balls, and here I am talking about blue balls again. The little blue balls are the phosphate head, and then the yellow tails are the fatty acid tails, all right? So the phosphate head is hydrophilic. It's happy to be in contact with water, so it faces the extracellular fluid, the water outside of the cell. And then the other phospholipid, the blue phosphate head, faces the intracellular fluid, the water inside the cell. The two sets of fatty acid tails face each other, and they create a very hydrophobic inner layer of the phospholipid bilayer. And that is what keeps water from getting in or from getting out, all right? That fatty boundary there. So some things do have to get in or out, and so there are various kinds of pumps and transporters that allow things to intentionally get in or outside of the cell. Ions in particular, what we need to look at for the action potential. So remember, ions, the main ions we need in this class: sodium, potassium, calcium, chloride. So we have channels that allow ions to go through. Remember, ions are charged particles and they can't diffuse through the phospholipid bilayer. Hydrophobic molecules can, very small molecules can diffuse across. Ions cannot because they're charged. So if we want ions to move back and forth across the cell membrane, we have to have channels for them. That's what ion channels do. So let's look at here's some illustrations. You can see on the far left side sort of a detailed illustration. Remember, these are proteins. Ion channels are big giant proteins. They're integral proteins that are embedded in the cell membrane. Proteins made of strings of amino acids, so that's what they're trying to show here. Amino acids, you have the amino group on the head, the carboxylic acid on the tail, and then remember, proteins have a primary structure, a secondary, a tertiary, a quaternary. Proteins fold up in 3 dimensions. So here on the far left, you're seeing that it's really just one long amino acid chain, but it has folded up to create these units, these subunits, that form an actual opening through the membrane. So second from the left, you see now a more schematized version of it, just showing that there are 4 components, 1, 2, 3, and 4. In the middle now, we're getting less technical. And finally, for E, they're just showing a damn pipe. It's like, the hell with showing any detail here. Let's just make it a pipe. Which it is. It's a pipe. That's how the ions get back and forth. So just get used to the various different cartoons of drawing it. The one on the far right is a little bit different kind of channel. We'll talk more about that later. But these are all ion channels. They allow ions to pass back and forth through the inside of the membrane. So just different artists are going to use different illustrations to do that. Okay? So, here we are. Here's a phospholipid bilayer. Here we see some integral proteins, some ion channels that are going through. They're passing through the membrane. They are allowing ions to flow through. So see the ions, little orange balls. You can see there's a closed ion channel and an open ion channel. When the ion channel is open, the ions can flow through. When the ion channel is closed, they can't flow through. What we're going to see is that in the nervous system, usually the ion channels are closed, but then something opens them. So at the neuromuscular junction, we saw that neurotransmitters opened them. Now we're going to see that electricity can open them as well. And actually, we did see that before. The calcium release channels in the terminal cisterns were voltage-gated channels. They were opened by electricity. So the flow of ions through channels creates electrical current. So remember, ions are electrically charged particles. Sodium +1, potassium +1, chloride -1, calcium +2. When they flow from one place to another, they are creating electrical current. This is real, actual electricity. It's the movement of electrical charges from one place to another, okay? That in turn creates a difference in the electrical potential between the two sides of the membrane. When charged particles move across the membrane, they're changing the overall charge on one side of the membrane versus the other. So now the membrane won't necessarily be at equilibrium. It may be more positive or more negative on one side or the other. And that's exactly how the nervous system works, by the difference in potential, in electrical potential on the two sides of the membrane. And that difference in electrical potential is created by ions flowing through ion channels and changing that electrical potential. So neurons are electrically excitable due to the voltage difference across the membrane. We'll see that the inside of the neuronal membrane, of the axonal membrane, is negatively charged compared to the outside, and that difference is what provides the impetus for the action potential. So let's take a close-up look at an ion channel here. What I want you to see is that ion channels don't necessarily just let any old ion through. They can be selective, okay? So ions will allow ions to pass from one side of the membrane to the other, but remember that the ion channel is a protein which is made up of amino acids. What can happen here is that the different sides of the protein, or different amino acids within the protein, can either face inward or face outward, and they can change their charges. So the amino group, the NH2, can sometimes pick up an extra hydrogen, become an NH3, and have a positive charge. In other cases, the carboxylic acid end of the amino acid can lose a hydrogen and have a charge of -1. So the amino acids themselves can be electrically charged. So according to the way that the long amino acid chain, the protein, according to the way that it folds up in the membrane, it may be that some of these amino acid charges are facing inside of the ion channel. So in this case, we see an ion channel that is folded up such that in 1, 2, 3, 4, 5 instances, the amino acid end of, or the NH2 end of the amino acid has picked up a hydrogen and it's now an NH3, so it's positively charged, okay? So there are 5 positively charged amino acids facing the inside of the channel. Now remember, what do we know about like charges versus opposite charges? Remember, like magnets, you ever play with magnets? If you get 2 magnets, you can turn the magnets so that they will attract one another and jump and, cement themselves together. Not cement, but bring themselves together. Or you can turn them so that they repel, right? So like poles repel, opposite poles attract, same as with charges. So like charges repel, opposite charges attract. So look what happens here. We have an ion channel, in this case, that has positively charged amino groups on the inside of the channel. Therefore, look what happens when a positive ion comes floating by. See up on the top, that positive ion came floating by? It's not going to be able to get through because all those positively charged amino groups inside the channel are going to repel it. But look what happens when a negative ion comes floating by. It actually gets sucked in. It's attracted to those positive charges. So this is how an ion channel can have a selectivity mechanism. Some ion channels will only let positively charged ions through. Others will only let negatively charged ions through. All right? That is the selectivity mechanism. Some ions get through, others are excluded. So, for example, would this channel be a chloride channel or a sodium channel? Remember, what charge does sodium have? What charge does chloride have? Sodium has a charge of +1, chloride has a charge of -1. So in this case, a sodium ion is going to get repelled, a chloride ion will get sucked through. So therefore, this would be a chloride ion channel, not a sodium ion channel. Do you see how that works? We're going to see that the action potential is controlled by having channels that are selective for certain ions, and that's how it's going to work. So here's how channels can be selective. This would be a chloride channel. Okay, so remember from Bio 156 again, I hate to keep saying that, but it keeps being true. Remember the sodium-potassium pump. All right, so sodium-potassium pump, also known as the Na plus K plus ATPase, moves 3 sodiums out as it moves 2 potassiums in. It's an example of active transports. Remember there was diffusion, passive transport, active transport. That was all in Bio 156. So what happens here is that this is a pump. It's an ATPase. Remember, anything that ends in ase is an enzyme. So this is an enzyme that, and why is it called an ATPase? Because it costs 1 ATP to use this enzyme. Every time it does its job, you have to spend an ATP. So what is its job? It takes 3 sodiums from inside the cell and it pumps them to the outside. It takes them from the cytosol, you can see in the lower left, and it pumps them out into the extracellular fluid. At the same time, it grabs 2 potassiums from the extracellular fluid and pumps them into the cytosol. All right? So burns an ATP for every exchange. The sodium-potassium pump burns 40% of your body's ATP at rest. Think about that. Holy smoke. You know, they say you need like 2,000 calories a day. Well, 40% of that, 800 calories. You spend 800 calories a day just powering the sodium-potassium pump. So what's going to happen as a result? Now, the sodium-potassium pumps, by the way, are everywhere throughout your body. They're in membranes all through your body, everywhere. Membranes all through your body have sodium-potassium pumps. They are constantly pumping sodium into the ECF and potassium into the ICF. So therefore, what's going to happen? It means that the extracellular fluid, the water outside the cell, is going to have more sodiums. And the cytosol, the intracellular fluid, the fluid inside, the water inside the cell, is going to have a lot more potassiums. This is going to create differences in concentration gradients. Remember? Concentration gradients. So if there are more sodiums outside, then sodiums sort of want to come in. Of course, ions don't really want things. But the concentration gradient will be driving sodium inside the cell. Likewise, because it's pumping potassiums in, potassiums are going to be concentrated inside the cell, so they are going to want, so to speak, to flow outwards, okay? We say that this pump is electrogenic because it moves 3 sodiums out and it moves 2 potassiums in. So notice it's moving 3 positives out and it's moving only 2 positives in. Therefore, what's going to happen? The outside of the cell is going to be more positively charged, and the inside of the cell is going to be more negatively charged. I mentioned this before. The inside of the membrane on an axon is going to be relatively negative. The outside is going to be relatively positive. So the sodium-potassium pump creates the concentration and electrical gradients that are going to allow the action potential to work. This is essential for the action potential. It's essential for your nervous system. Your nervous system can only work because of the sodium-potassium pump, which is always working. The sodium-potassium pump is working 24/7, 365. It is always pumping sodium out of the cell and pumping potassium into the cell. I say that the sodium-potassium pump is like the employees at Nicos, all right? 24/7, 365. You can go to Nicos anytime. You've been drinking margaritas all night. You need a carne asada at 3 a.m. Nicos has got one. One down there right by Prince and Campbell. I've been down there many times. By the way, always make sure, be nice to the people who work at Nicos, all right? They've usually got a tip jar right outside the window. Always put your change in the tip jar. Come on, they're sending money home to their families in Mexico. They're working hard, all right? One night I actually went into the Nicos, and they recognized me. I mean, I'd been there so many times. And they were all wearing their Nicos shirts. I said, whoa, those Nicos shirts are cool. Do you guys sell those? And they said, nah, we don't sell them. I said, that's too bad, because if you sold one, I'd buy one. And the guy I was talking to kind of like looked over his shoulder, and then he put up a little finger to his lips. He went, shh. He said, what size do you wear? I said, large. He said, okay. went in the back and got me one, see? So always tip the Nicos people. Maybe they'll give you a free shirt. You never know. Okay, so let's take a look at concentration gradients. So concentrations of ions is different inside and outside. So as we said, the sodium-potassium pump is pumping the sodiums out. Now chloride always goes where sodium go. This will pay you back. Remember this for Bio 202. Sodium chloride always follows sodium. So outside the cell membrane, there's going to be lots of sodiums. There's going to be lots of chlorides as well. Inside the membrane, there's going to be lots of potassiums. And also, just due to the nature of the inside of the cell, we talk about this a lot more in Bio 202. Cells make proteins. That's one of the jobs of cells. So there are lots of proteins inside the cell. And then the phosphate ion serves as a buffer. It helps control the acid-base balance inside the cell. So the inside of the cell has lots of positively charged potassiums and proteins. It has negatively charged phosphate ions. The outside of the cell has a lot of positive sodiums and a lot of negative chlorides. So notice there's both positives and negatives inside and outside the cell. Overall, the positives and negatives are relatively equally balanced. It's just that right next to the membrane, the inside of the cell is relatively more negative and the outside of the cell is relatively more positive. So the cytosol, again, full of potassium and proteins. The ECF, the outside water, is mostly sodiums and chlorides. The result is a concentration gradient for the ions. The two concentration gradients that are the biggest deal, the most important ones, are the concentration gradients for sodium and potassium. That's what's going to drive the action potential. So remember, the sodium-potassium pump has moved all the sodiums out. Well, not all of them. Moved a whole bunch of them out. It's moved a whole bunch of potassiums in. So now the sodiums are like just itching to get inside, and the potassiums are just itching to get outside. That's what a concentration gradient means, all right? It means that if there's ever an opening in the membrane, the sodium ions are going to come flowing from out to in, and the potassiums are going to flow from in to out, okay? That's created by that sodium-potassium pump. So that's the concentration gradient. There's also an electrical gradient. So remember that we were pumping 3 sodiums out, 3 positives went out, only 2 positives went in. That means there's relatively more positives outside the cell membrane and relatively more negatives inside. Therefore, the inside of the cell membrane has a relatively negative charge, okay? So that's what I'm saying there. The inside is more negative, the outside is more positive. That creates an electrical gradient. So remember about like charges and opposite charges? Because the inside is more negative, it means that positive ions are trying to get in. And because the outside is more positive, it means negative ions are trying to get out. The potential difference here is negative 70 millivolts. So negative 70 millivolts, you're thinking, wait, what? How do we get that number? Where does that come from? What's a millivolt? All right, think of the little, AA or AAA batteries you use in the remote control for the TV. Everybody's suddenly going, oh, yeah, the TV. Why didn't you say so? So, those little tiny batteries that you use in your TV, do you know how many volts those are? Do you remember how many volts are those little batteries that you use? Think about that. Go get one if you have to. You can look it up for yourself. But those are in whole volts. These are millivolts. So these are very, very tiny. All right? Millivolts are tiny. So the electrical charge here is minuscule, but it's just right for a cell. Okay? So the inside is relatively negative 70 millivolts. Now, why is it negative? We've been saying that before. Because we pumped more positives out. 3 sodiums went out, only 2 potassiums went in. That means the outside is relatively more positive. The inside is relatively more negative. So that's why it's negative. Why is it exactly negative 70? Well, we could use an equation, and I could run through the equation and show you, but it would probably put you to sleep. But it just turns out that due to the concentrations of ions in the human body, the number turns out to be 70. So it's negative 70 millivolts. That is a number I want you to know, okay? We call that the resting membrane potential. The resting membrane potential is negative 70 millivolts. So we could have measured the potential either inside or outside. If we measured it outside, it would be positive 70 millivolts. But as it turns out, it's easier for purposes of measurement to measure inside the cell. So that's why we go with negative 70 millivolts. We call that the resting membrane potential. The way to think about that is just that the cell membrane normally has a charge of negative 70, all right? We say the cell membrane is polarized. That means it has a different electrical charge on the two sides. It's not at equilibrium, okay? That creates the electrical gradient, okay? The inside is more negative, the outside is more positive. And once again, that's created because of the sodium-potassium pump. So we can take a look here. This is showing actual membrane or ion concentrations inside the human body in the upper left. You see there? Sodium outside the membrane is at 150 millimoles. Inside it's only 15 millimoles. So a lot more sodium outside. And then look at potassium. A whole lot more potassium inside, 150 millimoles inside. Potassium only 5 millimoles outside. You can see the chloride difference again. And then you can see that again the charge across the membrane is negative 70 millivolts. Okay? All right. So the electrical gradient, we're calling it a difference in electrical charge. The electrical gradient is the inside is negative relative to the outside. We say the resting membrane potential, negative 70 millivolts. The concentration gradient, the inside of the membrane has more potassium, the outside has more sodium. All right? So let's test and see if this came through. When the membrane is at rest, the concentration gradient pushes sodium where and pushes potassium where? So I'm asking about the concentration gradient. So remember, there's more sodium outside, there's more potassium inside. So where is the concentration gradient trying to get sodium to go? Remember, ions, particles, always move from areas of high concentration to low concentration. So since there's a lot more sodium outside the cell, sodium's going to try to flow from high concentration to low, and potassium likewise is going to flow from high to low. There's more potassium inside. So what ends up happening? Yeah, the concentration gradient is trying to push sodium inside. It's trying to push potassium outside. Okay? The concentration gradient means that because there's more sodium outside the cell, sodium will be trying to get in, and because there's more potassium inside, potassium will be trying to get out. Now, let's change the question a little bit. When the membrane is at rest, the electrical gradient pushes sodium where and potassium where? Are you with me? The electrical gradient, not talking about concentration gradient anymore, talking about electrical gradient. So where is the electrical gradient? So remember, like charges repel, opposite charges attract. So the inside of the membrane is more negative, the outside is more positive. Notice that both sodium and potassium are positively charged, so where do both of them want to go? Both of them want to go inside. So notice what happened here. Both the concentration and the electrical gradient are trying to get sodium to go in. But potassium has more of a push-pull kind of a relationship. The concentration gradient is pushing potassium out. The electrical gradient is pulling potassium in. So potassium's sort of more ambivalent. The sodium really has two powerful forces both pulling it in. Potassium has one force pushing it out, the other force pulling it in.